Welcome to the Deep Dive, the place where we take complex research and stacks of scientific papers and really just distill them into clear, actionable knowledge for you.
Exactly.
Our mission today takes us right to the cutting edge of human potential. We're talking about stem cell therapy.
A huge topic, and a confusing one for many people.
Oh, absolutely. It's a field that is, often shrouded in confusing claims, regulatory ambiguity, and frankly, a lot of outdated ethical debates.
Right. But the actual biology behind it is proving to be, I think, the most profound tool we have for true regenerative and anti-aging medicine.
Is a phenomenal area of science, but you are right, the complexity is high.
It really is. And for you, the listener, trying to sort the fact from, the hype, it's essential to understand that what we're really dealing with is the body's fundamental innate ability to heal itself.
Right, but we need to know what controls that process.
And how we can effectively augment it when the body's natural reserves start to decline. That's the key.
And we have an immense body of work anchoring this deep dive. It's centered around the clinical and scientific insights of a really remarkable expert, Dr. Joy Kong.
Her background is incredible. It connects dots that most researchers just don't even see.
It's wild. Triple board certified, psychiatry, neurology, addiction medicine, and anti-aging regenerative medicine.
So when someone bridges the nervous system, cellular aging, and addiction recovery,
Yeah.
the view is comprehensive.
Totally. She was recognized as the stem cell doctor of the decade back in 2021, and her commitment to evidence-based efficacy, that really forms the backbone of our source material today.
And that holistic view is exactly what's needed because stem cell action is systemic. It's not a single drug for a single symptom.
Right.
So our mission today is to cut through all that noise and provide some clarity. We're going to define the science of stem cells, settle some of those major ethical debates, and then, and this is critical, we're going to dive deep into the evidence that proves why the source of the cell is the single greatest predictor of therapeutic success and safety.
Okay, let's unpack this. So if we're going to understand how we can, repair a 50-year-old body, we have to start at the ultimate 0 point of potential.
You have to.
Where does the life of the stem cell begin, and what are the initial rules of, I guess, differentiation?
The story begins with the fertilized egg, that single cell.
Just one?
That is the quintessential stem cell. It's the definition of an all-powerful cell. In biological terms, we call it totipotent.
Totipotent, so total potential.
Literally total potential. It has the ability to become every single cell type in a human body, plus the placenta, the whole support structure. It can create a full, complete human being.
Wow. And as soon as that one cell starts to divide, that potential starts to narrow, right? It feels like a trade-off. You get specialization, but you lose that flexibility.
Precisely. That's a perfect way to put it. As the cell divides, the subsequent generations start moving down a pathway of specialization.
Yeah.
It's a process we call differentiation.
Like a river branching out.
Exactly like a river that branches into smaller and smaller streams. Once that first split happens, the cells become pluripotent.
Okay, so from totipotent to pluripotent.
Right. Pluripotent means they can still make any tissue type, brain, bone, heart, you name it. But they've lost the ability to create those external structures, like the placenta. This is the stage of the embryonic stem cell, or ESC.
Which we will definitely circle back to.
Oh, yes. And then you just keep moving down that path further and further down the river until you hit the final destination.
End of the line.
Exactly. You eventually reach what we call the terminal cells. These are the mature, specialized cells that make up your specific organs.
The final liver cell, the fully formed neuron.
The skin cell, yep. They have gained 100% of their functional role, but they've lost almost all of their regenerative potential. They can't become anything else.
No, I thought we had stem cells in our organs, though.
We do, and that's an important point. Within most organs, we still retain some local stem cells like in the liver or the gut. But their potential is what we call multipotent, meaning they are highly restricted.
So a liver stem cell can only
become a liver cell. It can't spontaneously decide to become a skin cell. Its fate is sealed, so to speak.
So the magic we're chasing in regenerative medicine, it lies in finding the cells that are far enough down the path to be safe and regulated.
But still flexible enough to be a versatile repair crew.
That's the sweet spot.
That is absolutely the sweet spot. That brings us to the two key types we encounter in research and therapy. First, you have those embryonic stem cells, the ESCs.
Right.
As we said, these are pluripotent. They're obtained very early, around day 5 to 7 after fertilization, when this little inner cell mass forms. Their sheer potential, the ability to make anything, is why they are so desirable for research.
But that potential is also their downfall, clinically speaking.
It is. It's the ultimate risk management problem. When you introduce them therapeutically, their lack of self-regulation means there's a very high risk of causing uncontrolled, disorganized growth.
Tumors.
Specifically, a type of tumor called a teratoma. These are just growths that often contain a chaotic mix of tissues. You'll see bone, hair, teeth, cartilage all jumbled together.
Because the cells are just too primitive to follow instructions.
They're too powerful, they have maximum potential, and with that comes maximum risk.
Which brings us to the workhorse, the one that's actually being used in clinics today.
Yes. The most utilized and, frankly, the safest cell type today is the mesenchymal stem cell, or MSC.
MSC.
These are multipotent cells that exist in various tissues. You find them in fat, bone marrow, and, crucially for our discussion, umbilical cord tissue.
And they're in between those other two types.
They're right in that sweet spot we talked about. They're specialized enough to be safe, but still flexible enough to become bone, cartilage, muscle, or fat. And most importantly, they are master coordinators of healing.
So what defines an MSC, no matter where it comes from? What's its core job description?
The fundamental defining feature that all stem cells share, but which MSCs perform so elegantly, is this ability to sustain their own population while also providing for repair.
How does that work?
They can do one of two things. They can replicate themselves, just make an identical copy to replenish the local stock, or they can differentiate.
Which is making a copy plus something else.
Right. They make a copy of themselves plus a more specialized daughter cell to go and repair damaged tissue. They're the essential sorts of replenishment and systemic repair signals.
Okay, so this brings us directly to the part of the conversation that, historically causes the most friction for listeners, the ethics, the legal landscape.
Right.
When people hear stem cells, they often conjure up these images of really highly charged controversies. Let's just immediately clarify the misinformation.
We have to. The initial high-profile controversy, the one that dominated the news for years, was entirely focused on embryonic stem cells, the ESCs.
The ones with the highest risk.
And the ones that required destroying an embryo to obtain. It all stems from that philosophical debate about when human life begins.
And that led to the George Bush ban.
It led to President George Bush's famous 2001 executive order, which, to be precise, halted federal funding for creating new ESC lines. It did allow research on existing lines to continue.
But that entire debate, as foundational as it was, is it even relevant to what we're talking about today?
It's largely irrelevant to modern FDA-compliant regenerative therapy.
And we have to be crystal clear about the difference between that past controversy and what's being practiced now with ethically sourced materials.
Absolutely. The confusion often blends ESCs with reports of, truly horrific, questionable practices that happened in other nations. There was a brief dark period where fetal tissue was obtained unethically in some Eastern European countries.
But that's not what's happening here.
We have to stress that is not what is happening in the United States in accredited regenerative practices. We are talking about umbilical cord-derived cells.
Which completely removes the ethical controversy.
The source is the key. Umbilical cord stem cells are obtained at the time of birth. Once the baby is born, separated, and healthy, the umbilical cord and the placenta are just biological waste.
They'd be thrown away otherwise.
Exactly. They are collected in a saline bag, put on ice, and shipped to a lab for processing. The crucial point for you to understand is this. The baby has been delivered. The tissue is already separated. No one is harmed.
The ethical argument about destroying an embryo is just. It's not applicable.
It does not apply here. And that clarity is essential for anyone trying to assess the morality of modern regenerative practice.
So now that we've established the ethical sourcing, let's talk about the regulatory friction that exists even when the sourcing is completely non-controversial.
Yes.
The ongoing conflict between the FDA and doctors is critical to understanding the current practice environment.
This is a major area of difficulty and misunderstanding. The friction really boils down to classification. How do you classify a stem cell?
The FDA's position is that it's a drug.
Their default position is to regulate stem cells as a drug. And if it's a drug, it falls under Section 351 of the Public Health Service Act.
Which means?
It means you need phase 1, 2, and 3 clinical trials, a multi-billion-dollar, decade-long process.
Wow.
The FDA requires this rigorous process because they view the cells, once they're processed and cultured, as a manufactured pharmaceutical product. They're concerned with purity, sterility, mass production standards.
But the practitioners, the doctors on the ground, they're pushing back on that fundamental classification. They don't see it as a drug.
They see it as human tissue, a natural biological material that is simply being transplanted or infused back into the body.
Like an organ transplant.
Exactly. They argue it should fall under Section 361 of that same act, which governs human cells, tissues, and cellular and tissue-based products,
Yeah.
HCTPs.
And that's the framework for
Organ transplantation, blood transfusions, skin grafts, things that have historically been governed by the practice of medicine, subject to medical board oversight, not classified as manufactured pharmaceuticals.
The analogy feels really strong. I mean, if a doctor removes your blood, processes it, and gives it back to you, that's just standard medical practice.
It is.
Why should stem cells from birth tissue be fundamentally different?
The difference in the FDA's view lies in a clause, the minimal manipulation clause.
Okay, what does that mean?
If you say, harvest bone marrow, and immediately inject it right back into the patient, that's considered minimal manipulation, and it's generally acceptable under that 361 framework.
But that's not what's happening with the umbilical cord cells.
No. If you take birth tissue, isolate the stem cells, culture them, expand them, test them, freeze them, the FDA argues that this extensive processing crosses a line. It becomes more than minimal manipulation.
And that transforms it into a 351 product, into a drug.
In their eyes, yes.
Yeah.
Which then leads to this whole practice of off-label use.
Which is actually really common in medicine.
Oh, incredibly common. 80% of medicine is practiced this way, often using approved drugs for non-approved conditions.
So in this context, doctors are arguing that using these cells to augment the body's own repair capacity is within their medical judgment.
Just like using a blood transfusion to support a patient after trauma. And this complex regulatory environment is why it's so vital for you, the listener, to understand the underlying science. Because when the government hasn't fully sanctioned the application, the onus is entirely on the physician and the patient to ensure the highest quality, most scientifically sound material is being used.
And that brings us right to the core question of quality control. What source actually works best? This is where we shift from theory to efficacy. And it gets really interesting here. We have three big camps for mesenchymal stem cells, MSCs. You have your own fat, your own bone marrow, also autologous or healthy donor umbilical cord-derived cells, allogeneic.
Correct.
So let's be provocative here. We have to address the industry that relies on using your own cells. What specifically is the most compelling biological evidence against using your own, say, 54-year-old repair crew?
The evidence is overwhelming, and it centers entirely on one factor: chronological age.
The age of the cells matters that much.
It's everything. When we use autologous cells, whether from fat or bone marrow, we are transplanting cells that carry the exact age, the exact history, and all the accumulated baggage of the recipient. The research comparing these sources, like the work compiled by Dr. Kong and her peers, it just reveals the undeniable superiority of the younger cells. They are consistently more potent and more vibrant.
So how was that superiority measured in the lab? I mean, what were the specific parameters that gave the cord cells the edge?
Well, researchers looked at the most fundamental metrics of cellular vigor. First, telomere length.
The little protective caps on the ends of our DNA?
Exactly. Shorter telomeres mean older cells. The umbilical cord cells, being brand new, have maximum length. Then you have mitochondria health.
The cellular powerhouses.
Right. Young cells have robust, highly efficient mitochondria. Older cells have sluggish, fragmented ones. They also looked at the growth factor profiles.
What healing signals they release.
And the younger cells secrete a vastly richer, more complex array of anti-inflammatory and regenerative molecules. They also looked at the rate of senescence, how quickly the cells age and die in a lab dish.
And the older cells just gave up faster.
Much faster. And finally, differentiation potentials. While they're all MSCs, the younger cord cells just retain a slightly broader range of potential, and they're faster and more efficient at turning into the target tissue, like cartilage.
So if I'm 54 and my own cells have accumulated decades of damage, what is that damage exactly? What is the baggage they're carrying?
Think of it as cellular pollution and physical exhaustion. Your cells have spent 54 years fighting off inflammation, environmental insults like sun exposure and pollutants, free radicals, oxidative damage.
Just the wear and tear of living.
The wear and tear of living. And this results in the accumulation of two major problems.
Yeah.
First is misfolded protein buildup.
So proteins that are the wrong shape and they just clog things up.
They clog up the cell's machinery. The second is lipofusin accumulation. This is often called the age pigment. It's a byproduct of cellular breakdown and waste that the cell just can't clear effectively anymore.
So it's just full of junk.
It leads to sluggish function. So when you harvest a 54-year-old stem cell, you are harvesting a cell that is already fatigued, compromised, and has damaged DNA. Asking it to repair a 54-year-old body is like sending a tired, injured soldier back to the front lines.
I remember the old marketing argument, though, particularly for the fat-derived stem cells. The idea was that they were somehow dormant and preserved in the adipose tissue,
Right.
that they were just tucked away, pristine, and immune to the aging process.
That notion is scientifically unsupported. It's counterintuitive to everything we know about biology.
Why?
Stem cells residing in fat adipose-derived stem cells, they're metabolically alive and active. They're constantly reacting to the local environment, which includes all the toxins, the inflammatory signals, and the oxidative stress circulating in your body.
So they're not sealed off.
Not at all. I mean, think about it. If the cells that create eggs and sperm, the germline cells, decline rapidly with age, why on earth would the fat stem cells, which are constantly interacting with this toxic environment, remain magically pristine? They don't. They carry the age of the host. Full stop.
Okay, but now we have to address the biggest challenge to the donor cell approach, and that's rejection.
Of course.
If I introduce young cells from a newborn's umbilical cord, that's foreign tissue, how does my immune system not launch a massive attack and just reject all of them? That seems like a huge trade-off for getting more potent cells.
That is a fantastic question, and it highlights the single most important biological trait of mesenchymal stem cells. They are immune privileged.
Immune privileged, what does that mean?
Unlike most cells in your body, MSCs have a very low expression of major histocompatibility complex proteins on their surface, MHC proteins.
And those are the flags that cells use to identify themselves to the immune system.
Exactly. They're the flags that say, I belong here, or I'm a foreign invader.
So if the flags aren't flying very high,
The immune system doesn't really see them as a threat. They are essentially stealth cells. Because they're not strongly expressing those I am foreign flags, your T cells don't immediately recognize them as allogeneic, as foreign, and launch a rapid rejection response.
but it's more than just hiding.
Oh, much more. Young MSCs are profoundly immunomodulatory. They don't just hide, they actively secrete anti-inflammatory cytokines that calm down the local immune response.
So they're like diplomats.
They essentially tell the recipient's immune cells, stand down, we're here to help, not invade. They create their own little pocket of peace so they can get their work done.
So the body lets them do their work, transfer all those healthy components, launch the repair campaign, and then they eventually naturally degrade after a few months.
That's the mechanism. This immune privilege is what makes the allogeneic umbilical cord source not just feasible, but highly effective. You get the maximum vibrancy without that biological safety trade-off of rejection.
And this age factor, it leads directly into the safety discussion, particularly around cancer risk. This is an insight that's genuinely transformative. It changes the discussion from just cell replacement to something like cellular intelligence.
This is a core finding from the research.
Yeah.
Older autologous stem cells, because of their cumulative damage and aging, what Dr. Kong aptly calls a form of cellular dementia, they lose their ability to accurately tell friend from foe.
Cellular dementia, I like that. They get confused.
They do. They become indiscriminate growth promoters. So if you have an existing small cluster of cancerous or precancerous cells that you don't even know about,
Which many of us probably do.
right, an infusion of these damaged older MSCs can potentially give those cancer cells the grow, grow, grow signal. It's important to note, they won't create new cancer, but they can act as an accelerant for existing subclinical disease.
The younger cells, though, they retain the sophistication to act more like a quality control manager.
That's the difference. Umbilical cord-derived stem cells are smarter. They retain their full signaling capability to assess the local environment. There are studies comparing them directly next to aggressive cancers, like glioblastoma.
A really nasty brain cancer.
Very nasty.
Right.
And they showed that while the older autologous cells blindly promoted growth, the younger cord cells demonstrated the ability to discern that the cancer cells were aberrant.
Aberrant meaning just wrong, not behaving correctly.
Exactly, not behaving correctly. And they then sent signals, cytokines and exosomes, to the surrounding tissue and immune cells to either initiate apoptosis
Program cell death.
in the cancer cells or to trigger the immune system to come in and get rid of them. They launch an anti-cancer, anti-aberrant cell campaign.
That distinction, it fundamentally changes the risk-benefit analysis. We're not just choosing the most potent cells, we're choosing the safest, most discerning ones.
And that's a critical point for anyone considering this therapy. So moving past the source comparison, we now look at how MSCs actually function once they're infused. And this is where the old paradigm, the idea that stem cells just become replacement tissue, is completely shattered.
Right, that's not their main job.
Their primary job is not replacement, it is systemic command and control. Dr. Kong refers to them as the body's smart pharmacy.
The smart pharmacy.
They are constantly monitoring the entire system, sensing local inflammation, fibrosis, or injury, and then they compound the exact cocktail of healing signals needed for that specific problem.
And this smart pharmacy, it relies on a healthy, robust stock of cells that are already in our body. But that stock is rapidly depleted with age.
It is.
Yeah.
We have to first understand where our native MSCs are coming from. Many of them are derived from cells called parasites.
Parasite.
Parasites are these specialized cells that literally cuddle around the walls of our blood vessels. Think of them as dormant sentinels, or like a repair crew hiding in the building's infrastructure.
Waiting for the alarm to go off.
Exactly. When damage occurs, whether it's a cut, a heart attack, or chronic inflammation, these parasites detach, they become active MSCs, and they migrate to the site of damage to orchestrate the repair.
And that capacity, the capacity of that repair crew is what we lose as we get older. You have the data on this decline, and it's pretty shocking.
The statistics are staggering, and they explain why our repair capacity slows so dramatically. The concentration of these vital parasites, these MSCs, it just drops precipitously.
So what are the numbers?
At birth, 1 in every 10,000 cells is an MSC.
1 in 10,000. Oh.
By your teenage years, it's 1 in 100,000.
Wow, that's a tenfold loss of your command and control capacity already as a teenager.
A tenfold loss. By your 40s, it's 1 in 400,000. And by your 80s, it plummets to 1 in 2 million cells.
1 in 2 million, it's almost gone.
It's a tiny fraction of what you started with.
And that huge drop in the ability to generate these healing signals is what drives aging, often called inflammaging. We should probably define that term clearly for the listener.
Right. Inflammaging is chronic, sterile, low-grade systemic inflammation. It pervades the entire body, and it is the single biggest driver of age-related disease.
And when you say sterile, you mean it's not caused by a bug, like a virus or bacteria?
Correct. It's not caused by an acute infection. It's driven by the accumulation of damaged, aged, or senescent cells, including the aged stem cells themselves.
So when our own stem cells acquire that cellular dementia we talked about, they actually become part of the problem.
They become a huge part of the problem. When stem cells become senescent, they're not just dormant or useless, they become toxic.
How so?
They start secreting a destructive cocktail of inflammatory molecules. It's called the senescence-associated secretory phenotype, or SASP.
ASP.
And these inflammatory cytokines, things like interleukin-6 and TNF-alpha, they destroy healthy neighboring cells, they drive chronic fibrosis or scarring, and they accelerate the aging process across every single organ system.
So the body gets trapped in this vicious cycle.
Exactly. The repair crew is too depleted to fix the damage, and the remaining crew is starting to sabotage the whole system.
Which means that by introducing young, vibrant MSCs, we're doing a lot more than just local repair. We're effectively turning down the volume on that chronic destructive inflammation.
The ability to reduce inflammaging is the cornerstone of anti-aging medicine. I mean, aging is the biggest risk factor for almost every chronic disease.
Heart disease, neurodegeneration, diabetes.
All of it. So if we can significantly slow or even reverse biological age, the impact on public health is just immense. Consider this statistic. Reversing biological age by just 7 years is projected to cut the existing burden of chronic illness in the population by a staggering 50%.
50%. That's incredible.
It shows why rejuvenation isn't just about longevity. It's about disease prevention. It's about health span.
Okay, so let's move past the systemic inflammation reduction and into the specific, fascinating ways these young cells communicate their instructions.
Yes.
We mentioned that only a small percentage of the infused cells might stick around, yet the benefits can last for a year or two. How is that possible? It must require some kind of advanced cellular communication.
It does. It requires what we see as the 3 key mechanisms of systemic repair, which go far, far beyond simple tissue replacement. The infused cells act as powerful biological instructors, not just as building blocks.
Okay, mechanism 1, the transfer of raw energy.
This is the mitochondria transfer mechanism. It's a truly breathtaking piece of modern biology.
It sounds like science fiction.
It does. Aging cells suffer from mitochondrial decay. Their power plants become sluggish and inefficient. Young, healthy umbilical cord cells packed with robust mitochondria are capable of physically transferring these healthy powerhouses directly into the recipient's existing cell.
They just donate their batteries.
Think of it like a biological jumpstart. The young cell doesn't just replace the old sluggish cell, it gives the existing cell a brand new, fully charged battery pack. This instantly boosts its energy production and its functional horsepower.
Wow, okay, so that's mechanism one. What's mechanism two?
Mechanism 2 is all about the information packets that rewrite the software. This is the critical role of exosomes and microalign.
Exosomes, so these are the little delivery drones from the smart pharmacy.
That's the perfect analogy.
Yeah.
The stem cells produce millions of these tiny lipid-encased packets called exosomes. And inside them are growth factors, cytokines, and most importantly, microRNAs.
MicroRNAs. They're not genes that code for proteins.
No, they're sophisticated genetic regulators. They're like the software managers.
So how do they act as a genetic regulator? What's the analogy for fixing the foundation here?
Think of microRNAs as specialized software patches or genetic silencers. They can travel via the exosome into the nucleus of your existing cells.
Right into the command center.
Right inside. And once they're there, they can attach to specific messenger RNA sequences and literally repair the DNA programming. They can tell an aged, inflamed cell, turn off the old inflammatory software and turn on the young regenerative software.
Like telling it to make more collagen or to stop making scar tissue.
Exactly. They are reprogramming the recipient's cells to behave younger and healthier from the inside out.
That is profound, and it means the benefits persist long after the messenger cells are gone.
Absolutely. The stem cells themselves, whether they stick around for 1 month or 3 months, they have a limited lifespan. They're fighting battles, they're calming inflammation, they're differentiating.
They get used up.
They do. But the information they transferred, the repaired DNA, the renewed mitochondria, the silenced inflammatory pathways, that lasts. This is the underlying mechanism that explains why the clinical benefits, like reduced chronic pain or improved organ function, can last for a year or two down the line, even though the infused cell population has mostly expired.
The renovation is complete, so the construction crew can go home.
Exactly. The cells did their job by renovating the existing system.
And we have hard data showing this systemic reset, right?
We do, particularly from animal models that used repeated intravenous infusions. Researchers tracked key markers of aging and function. What did they find? They found that in the brain and muscle tissues of the receiving animals, neurotransmitter levels, growth factor profiles, and key senescent markers were all driven back to the younger state. The entire biological clock was effectively rewound by the infusion of these young signals. It just reinforces the idea that we're treating the root cause of aging, not just managing the symptoms.
Okay, let's transition now to the practical application of this powerful mechanism. We established the regulatory conflict earlier and the fact that most regenerative therapy is off-label.
Right?
So what does that look like in the clinical setting, and what are the established benefits that doctors are reporting?
First, we have to reiterate the legal distinction. The only FDA-approved indication for stem cells is hematopoietic reconstitution.
And let's quickly define that again. It's a very technical term.
Hematopoietic reconstitution simply means rebuilding the entire blood and immune system. It's a bone marrow transplant.
Usually after something like chemo has just wiped everything out.
Exactly. After intensive treatments like chemotherapy or radiation have completely destroyed the patient's existing immune system. It is a radical, life-saving, total system overhaul. It's a very different scale and mechanism from what we're discussing in regenerative anti-aging therapy.
Right. So outside of that very narrow application, we're relying on the off-label practice of medicine. The goal is augmentation. So what kind of serious, difficult-to-treat conditions are seeing success?
Clinically, doctors are augmenting the repair capacity for conditions where the body's natural repair mechanisms have just failed. We've seen reported benefits in notoriously difficult chronic conditions, things like COPD.
Chronic obstructive pulmonary disease.
Right. Stem cells can reduce the chronic inflammation and the fibrosis, the scarring, and the lungs, potentially leading to improved gas exchange. We're also seeing it used for liver cirrhosis.
So it can help the liver heal itself.
By reducing inflammation and promoting the activity of the local liver stem cells via those exosomes, they help the liver halt and sometimes even reverse scarring, and also cardiomyopathy.
A declining heart function.
Yes. The cells help repair damage and improve contractility by reducing inflammation and boosting that mitochondria health in the existing heart muscle cells.
So the goal then, it's not merely to sustain the current level of health, but to push for genuine reversal and healing. It's tapping into that phenomenon we sometimes hear about as spontaneous remission.
Exactly. Traditional medicine often focuses on managing the decline. Regenerative medicine focuses on facilitating the body's attempt to heal and repair itself, which often results in outcomes that were previously considered impossible.
Now, how do we best deliver this smart pharmacy? Does it make more sense to inject them locally right where the problem is, say, a damaged knee, or to infuse them systemically through an IV?
The optimal delivery method really depends on the condition. The intravenous or 5E approach is the most widely beneficial for systemic problems.
Like autoimmune disease.
Right. If you have autoimmune disease, high systemic inflammation, declining organ function in your heart, liver, lungs, kidneys, spleen, 4 is the logical choice.
But why does the IV approach work for a localized problem? Don't the cells just sort of circulate endlessly?
No, the body is smart. Diseased or inflamed areas express specific chemokine signals, or essentially chemical distress calls,
A flare gun.
a biological flare gun. And these distress calls actively attract the MSCs out of the bloodstream and toward the site of damage. So organs that are well perfused, meaning they have high blood flow, like the heart, liver, and lungs, are highly accessible to IV-infused cells.
But the large joints are a different story.
They are. For conditions like severe osteoarthritis in the knee, hip, or shoulder, a local injection is typically necessary.
Why is that?
The drained capsule is just not well perfused. The exchange of nutrients and blood products is very, very slow. It could take 2 months for an IV infusion to deliver an adequate therapeutic concentration to a large, compromised joint space
So direct injection gets them right where they need to be.
immediately. And similarly, some clinicians will use targeted delivery right into the spinal canal, which is called intrathecal, for CNS conditions.
Okay, so if my knee's the main issue, why would I even bother with the systemic IV approach? Why not just do the local injection?
Because there's a crucial trade-off.
Hmm.
While the local injection delivers a high initial concentration, the IV infusion offers a broad range of systemic benefit.
Ah.
Even if your primary complaint is the knee, you likely also have some subclinical inflammation in your liver, or you're developing atherosclerosis in your arteries. The IV approach engages the entire peripheral lymphoid system, your spleen, your lymph nodes, and it mounts a comprehensive anti-inflammatory campaign that a strictly local injection is going to miss.
So a combined approach is often the best.
Often the most comprehensive strategy, yes. You treat the local fire and the systemic kindling at the same time.
Let's talk about the anti-aging regimen itself. Using this treatment purely for rejuvenation and prevention, even when a person is already healthy. If the cells have a limited lifespan, how do you maintain the effect?
So we established that the cells live in the body for about 1 to 3 months, fighting battles, modulating the immune system.
Right.
And then the powerful downstream effect, driven by the exosomal communication and the DNA repair, that lasts for another 3 months or so.
So a total of maybe 6 months of benefit?
Roughly.
Huh.
Therefore, to ensure that your body is always operating under the instruction set of young, vibrant cells, the recommended anti-aging regimen is an infusion every 3 to 6 months. A 3-month schedule ensures that peak effect is maintained consistently.
Okay, this brings us to a really critical listener question.
Yeah.
For someone who is already proactively healthy, who's following this regimen, do you feel the difference every time?
Right.
Because if this is preventative medicine, how do we manage the expectation that it should feel like taking a stimulant or something?
This is a vital point for managing expectations. When you are already healthy, the profound benefits, the DNA repair, the reduction of chronic inflammation, the halting of senescent cell buildup, all of that is happening entirely under the radar.
So you don't feel it.
Most of the time, you don't feel a whole lot. If you're waiting for a dramatic surge of energy, you might be disappointed.
So how do you sustain the faith in the regimen then? What are the subtle signals that tell you the cellular repair is actually happening?
Well, the faith is grounded in the underlying science. Knowing that our MSC concentration is plunging as we age, and knowing the mechanisms of action, the mitochondria transfer, the microRNAs, are scientifically validated.
The logic is sound.
But you do sometimes catch subtle, specific cues. For me, sometimes it's a temporary feeling of heightened mental clarity or focus for a few days, or perhaps an improvement in sleep quality that lasts a few weeks. But crucially, what I notice is resilience.
Resilience.
I might notice that a heavy workout that used to leave me sore for 3 days now only leaves me sore for 1. Or I might notice that while everyone around me is getting the winter cold, I remain untouched.
So it's the absence of decline.
It's often the absence of decline, the improved recovery time, or the enhanced resistance to environmental stressors that confirms the systemic reboot is working. You are essentially protecting the body's biological reserves long before the symptoms of aging become overt.
That reframes the benefit completely. It's not a dramatic intervention. It's a foundational, quiet act of maintenance that prevents a future catastrophic failure.
Precisely. We are proactively addressing the decline in the MSC population before we hit that critical inflection point in our 40s where repair capacity really begins to lag behind damage accumulation.
Finally, let's explore the non-injectable localized applications, because this is often the entry point for listeners to regenerative medicine, topical creams, and skin health.
Right. The skin is our largest organ, and it is the most visible barometer of systemic aging and inflammation. Topical applications allow us to harness those regenerative signals without an infusion.
So what's in a high-quality cream like this?
It's formulated to deliver the beneficial components derived from umbilical cord stem cells, usually combined with supporting ingredients like peptides, antioxidants, and specialized natural oils.
Okay, to clarify the science here, you mentioned the cells in the topical cream are not alive, so what's actually doing the work?
The cells themselves are non-viable, correct. They are not living entities being integrated into your skin. However, the culture media they were grown in, which is known as the conditioned media, is a gold mine.
That's where all the good stuff is.
It contains all the potent biological signals they secreted while they were alive, the crucial growth factors, the cytokines, and those tiny information packets, the exosomes. All of those regenerative molecules are stable and, when they're formulated correctly, are very easily absorbable by the epidermal and dermal layers of the skin.
So we're delivering the smart pharmacy signals directly to the surface to address local aging. What are the confirmed measurable benefits in the skin studies?
The benefits are substantial, and they've been validated in controlled studies using various imaging techniques and sometimes even biopsies. The key functions are, first, collagen production.
Right, making the skin thicker and more elastic.
The growth factors signal the fibroblasts, the body's collagen-making machines, to ramp up production. Second is inflammation reduction. They calm down local inflammation, which is key to reducing redness, acne flare-ups, and sensitivity.
And then discoloration and fine lines.
By boosting cellular turnover and providing those DNA repair signals, they reduce the appearance of sun damage and smooth out fine lines.
So it's essentially applying the systemic repair instructions directly to the foundational structure of the skin.
Exactly. It uses the same biological mechanisms, signaling for repair, reducing inflammation, boosting local energy that the IV therapy uses systemically, but in a highly concentrated, localized manner for the largest, most exposed organ we have. So, to quickly bring our deep dive to a close here, the foundational truth of regenerative medicine is that younger cells are just superior.
No question.
By ethically sourcing vibrant, immune-privileged mesenchymal stem cells from umbilical cord tissue, we bypass the dangers and the limitations of our own aged cells. And this process initiates a system-wide reset. It reduces chronic inflammation, it repairs DNA through microRNAs, and it revitalizes our existing cells via mitochondria transfer.
So what does this all mean? It means we are crossing a vital threshold in medicine. We're moving beyond simply managing chronic disease into a phase of, quantified, achievable biological rejuvenation.
I think so.
We can now focus on resetting the health of the body at the foundational cellular level, pushing back against that inevitable biological decline that fuels most chronic illness.
And we touched briefly on one of the other essential ingredients in that topical formulation, peptides.
Right.
Peptides are these short chains of amino acids that act as extremely precise signaling molecules. They can prompt cells to do very specific things, like heal faster, build muscle, or increase certain hormones. When you combine them with the broader cellular signaling of stem cell components, the synergistic effect is immense.
Indeed. The world of regenerative and anti-aging benefits provided by peptides is a massive field of study in its own right. It's almost a parallel universe of rejuvenation potential.
It is. And the science is so deep that the conversation around that associated treatment modality continued for an additional 45 minutes beyond the source material we analyzed today.
We will definitely have to save that specific deep dive into peptides for another time. But for now, just consider the profound implications of this. Treatments that don't just patch up the symptoms you can see, but fundamentally reset the molecular machinery that governs your body's aging process. That foundational, proactive approach is truly revolutionary.